Okay, so welcome, students, members of the faculty. We are really pleased and honored of welcoming here Svante Pääbo, Princesa of Asturias Award for Scientific Research in 2018, Nobel Prize for Medicine and Physiology 2022 for, and I quote, the study of the genome of hominids and human evolution. This is a very, very special occasion for this faculty. And I guess this is the best lecture that we can have in this lecture hall. Just a few notes on what the contribution of Svante Pääbo has been to science, the enormous contribution of Svante Pääbo to science. It may be interesting that he started his PhD on viruses, not on human evolution, but at the same time he was studying so different subjects as Egyptology, Coptic language, Russian, and he started to study the DNA in mummies, in Egyptian mummies. And at that time, when he was a PhD, the development of molecular biology was still at the beginning, so the amount of DNA he was able to recover was not so much and was mixed with a lot of DNA from other species. But the history of his studies are the history of molecular biology in the late 20th century and the 21st century, because when he went as a postdoc to the United States, he became aware of PCR and the other methods and massive sequencing, so he was able to get much better samples of DNA from hominid species. When he came back to Europe, he founded in 1997 the Max Planck Institute for Evolutionary Anthropology, in which he did most of his research on ancient DNA from hominids. And he has studied the DNA of Neanderthals and the relationship of the presence of DNA in Neanderthals in current human populations. And he also has discovered a new hominid species, which is the Denisovans, from a very far location in the Altai Mountains in Asia. He has solved the problem with the relationship of ancient DNA from Neanderthals and has discovered that there is both replacement of mitochondrial DNA from Neanderthals coming into humans, and also assimilation to nuclear DNA in humans. As I said before, this is the best lecture that we can have in this faculty. His discoveries, apart from being a breakthrough in science, should show us humbly that as a species, we should not care about injustice, we should not care about frontiers, we should not care about differences in sex, in gender, in sexual orientation, in anything like that, because we are so similar that we are also closer to a different species as Neanderthals and Denisovans. So let me finish with a quote from Martin Luther King, when he wrote a letter from Birmingham jail, saying that injustice anywhere is a threat to justice everywhere. We are caught in an inescapable network of mutuality, tied in a single garment of destiny. Whatever affects one directly affects all indirectly. We are just one species, we are all humans, and please welcome Svante Pääbo and take the best of this lecture. Thank you.
Okay, it's a great pleasure to be here and see so many people in the room. It's a bit scary too. So I understood that we should try to have more of a dialogue and discussion. So I have, of course, slides that is sort of like a lecture, but maybe we stop here and there and see if we can sort of take questions now and again and see if we can have a bit of a discussion. Let me see. Or I can, that's a pointer, or I can turn this on, maybe.
Yeah, there's a switch.
There's a switch somewhere.
Okay, fine.
So just as a background then, I want to remind you about then what one has found when you study genetic variation worldwide, that I think you're aware of, and that is the fact that if you look at genetic variation, then most genetic variation exists in Africa today. And if you take the entire world outside Africa, although many, many more people live outside Africa than inside Africa, of course, there is yet less variation there. And the reason for this is that there is sort of a component of the variation inside Africa that doesn't exist outside Africa. And the interpretation of that is that modern humans, and with that we then mean the direct ancestors of everyone alive today on the planet, evolved in Africa, lived there for a while, accumulated genetic variation, which takes time, and then part of that variation went out and colonized the rest of the world. And we can use sort of genetic tricks to figure out approximately when that leaving Africa happened. And it's quite recently in evolutionary terms. It's sort of less than 100,000 years ago or so. Now, an interesting, and modern humans then evolved in Africa. The first things that sort of look like modern humans are something like 200,000, 300,000 years ago, so quite recently. Now, an interesting thing with that is that modern humans were not alone on the planet at that time. There were other forms of humans around. And most famously then, in Western Eurasia and in this part of the world, there were what we call Neanderthals. And in Eastern Eurasia, there were other forms of now extinct humans that we are beginning to learn a bit more about. In Africa, there were also other forms of humans that are not well known today. But so our group is then since 20-25 years sort of studying Neanderthals. And you may sort of wonder why we should be interested in Neanderthals who are not here anymore. I think it could be many reasons for that, but for me there are at least two reasons that makes me interested in it. And one is that Neanderthals are indeed the closest evolutionary relatives to everyone around today. So if we sort of want to define ourselves as a group or as a species, if you like, from a genetic or biological point of view, it's sort of them we should compare ourselves to and see in what ways are we similar to them, in what ways are we different from them. And of course, another thing that's interesting is that Neanderthals existed until about 40,000 years ago or so quite recently. So say, I don't know, hard to count, like 1,400 generations ago or so, there were still Neanderthals here. So it's sort of an interesting question is how did we interact with them, what happened when one met Neanderthals and so on. But to then do that, you need to, from a genetic perspective, you need to retrieve DNA from bones that are at least 40,000 years ago. And that's sort of what, over 40 years or so, our group has worked on. And that all started, as you heard, with ancient Egyptian mummies, which are not that old. They are 2,000, 3,000, 4,000 years old. They look very well preserved. So back in the 80s, we sort of thought maybe there's DNA surviving there. Although most people at the time thought that DNA was a very unstable molecules that would be degraded rapidly after death. But if one looks with histology back then, just in a microscope in the tissues, say in the skin of that mummy you looked at there, you can see things in the basal levels here that looks like cell nuclei, and you can also stain them for DNA. So there seems to be DNA there in the cell nuclei. So made me very excited, and I went on to extract the DNA from there, clone it in bacteria. So you have many bacterial colonies with different DNA fragments from that mummy in it. And among them, we found some that had allorepeats in them, which is a repetitive element that exists in primate genomes, in sort of 1 million copies or so in humans. So it was an easy way to show that, yes, there is some human DNA in there among those clones. And published that in prestigious journal, Nature, to great fanfare. And sort of thought at the time, wow, this is cool. We will sort of now solve everything. We will study population history over time. In Egypt, look at things like when Alexander the Great came and conquered Egypt. Was it a big population replacement, or what happened when the Romans came, etc. But it turned out to be not at all as easy as we thought. Sort of over the next couple of years, it became very clear that there are many, many technical problems with this. And it also became clear that there's a huge, huge problem with contamination of your experiments. So these DNA sequences I had published there, they are quite long. They are, 1,000 nucleotides, 1,500 nucleotides. And we have learned that such long fragments sort of never exist in all tissues. It's rather like 30, 40, 50 nucleotides. So those sequences I published there surely came from myself or from some museum curator or from the chemicals we used. So it's sort of interesting to think back and realize that it was sort of really very wrong. So sort of use that now as an example of how you can go wrong in science, but you still perhaps make a step forward. It's an interesting discussion. We could have that discussion. Should one sort of retract this old paper that everyone now still sort of thinks this was the beginning of the field or not? I don't know, but are there opinions on that? Anyone wants to suggest we should suggest retract it? I at least go around and always say that it was wrong. So, and the technical issues are then, as we heard, right, that there's very little DNA surviving in old tissues like that, millionfold less or so than in fresh bone, for example. It's degraded to short size. There are chemical modifications in the DNA that can give the wrong sequences when you sequence. The vast majority of the DNA is microbial, but since there is so little DNA there, even tiny amounts of DNA contaminating your experiments. That could be, say, in a room like this, much of the dust are actually skin fragments from people that have been in the room. And one such little dust particle can contain way more DNA than what you have in your DNA extract. So whereas that won't influence experiments at all if you study present-day DNA, it can be sort of totally overwhelm the things here, and that's sort of what happened in the beginning. So what came was a sort of a long period where we got more and more hysterical about using clean room conditions, using bleach to destroy all present-day DNA, testing all chemicals, etc., etc., and also working on extinct animals. Because it's, of course, much, much easier if you study a mammoth and find something elephant-like there, you're very sure you have the right things. If you study an Egyptian mummy and find something human-like, you still don't really know if it comes from you or from the mummy. So mammoths, say, and extract. And we worked on all steps on how you sort of extract things efficiently, handle these short fragments. And what, of course, also came around was the human genome. That was sort of the first version finished in 2003 or so. So that you could sort of begin to dream about, in the beginning of the millennium, to just extract the DNA from a bone, make a DNA library on it for high-throughput sequencing, where you just ligate on things on the end and just sequence everything that's in there, rather than trying to retrieve, like PCR, as we heard, a certain fragment, part of the genome or so. And we got better at all of that, things got more efficient. And the cool thing here, when you sort of now sequence fragments like this is that you see the extreme ends of them. If you do PCR, right, you have to make primers in there and you never see the ends of the molecules. But here you see the ends, and there we sort of discovered something strange. That if we look at Cs, cytosine residues toward the end, they really appear at Ts, many of them. So there are many, like 50% or so of the Cs look like they mutated to a T. So that can be a problem. It's sort of hard to know what if it's a C or a T there. But the good thing is that one knows now why this occurs. It's a chemical reaction where the cytosine loses this amino group here. It becomes a uracil. That's otherwise a base that's just in RNA. That shouldn't be in DNA. And when you use enzymes to sequence, this is read as a T. So that sort of results in these apparent C to Ts. And the cool thing is that that takes hundreds of thousands of years of this reaction to happen. So for the molecules where you see the C to T changes in the end, you're sort of very sure that they are really old. So you can even restrict your analysis on only those molecules, for example, in an extract that has C to T in the end. And then be very sure that you study ancient things. And when I've done this from lots of extinct animals and things like that, but we are really into Neanderthals then. And we spent quite a lot of time then in the early part of the century to look for good sites where there's good preservation of Neanderthal bones and DNA in the bones. And the best site we actually found was in Croatia, this Vindia site, and we used 3 different bones at the time and sequenced then over 1 billion molecules and mapped them to the human genome, taking these C to T changes in the ends into account. And by 2010 we then had one-fold coverage, as we say, which means on average each position in the human genome was covered by 1 fragment. That still means that many parts are covered by, say, 2 fragments, and we miss parts, so we have about half of the genome we had back then covered. But we could begin to ask questions. And how should we make this into a discussion, actually? I'm just going on like a German professor that never stops speaking. I don't know. I hope someone dares interrupt me and sort of when I'm unclear or so we sort of...
That's unlikely.
I don't know. But sort of the first question we were interested in back then was this relationship of Neanderthals to present-day people. Because there were really a lot of opinions there among paleontologists about that. I don't know how many paleontologists are here and know what I dare say, but there were 2 extreme views. One is this, that modern humans come out of Africa and replace all other forms without any admixture. So Neanderthals have no relationship to anybody living today, nor, say, forms of humans in Asia, and so on. Another really extreme view that probably no one really had back at that time is that the Neanderthals are the ancestors of Europeans. That Europeans today would be modern-day Neanderthals or have a big contribution, more likely genetically, from Neanderthals. And we had earlier, I didn't have those slides here, but the first part we got of the Neanderthal genome was the mitochondrial genome. That is sort of many copies per cell, so it's particularly easy to retrieve. And it's inherited from mother to offspring, so you sort of look at the female side of history. And that was very clear that the Neanderthal mitochondrial genome was very different from present-day people, fall outside the variation. So if you look at the mitochondrial genome of everyone on the planet today, humans, you go back to an ancestor 100,000 years ago, 150,000 years ago. And the Neanderthals sort of go back at least 500,000 years or so. So it sort of fell outside. And that had really biased our thinking, at least. We sort of I'd wondered if there is contribution from Neanderthals. We found no contribution at all in the mitochondrial genome. If you read our papers, we even say the situation might be different for the nuclear genome, but we really sort of thought, since we see nothing from the mitochondrial genome, there is probably no contribution. But so when we began to get now the nuclear genome, we sort of started out by asking this question, is there a contribution to Europeans? And if that was the case, we would of course expect Europeans today to carry genetic variants that are related to Neanderthals, and that Europeans should do that more than other parts of the people in other parts of the world, particularly in Africa. Since Neanderthals have never been in Africa, there's no reason to assume that Africans should have anything special to do with Neanderthals. And we put together a consortium of population geneticists and people to help us with this. We had this Croatian Neanderthal sequence there, and we went out and sequenced 5 people from different parts of the world. And at that time, one was sort of genome sequencing was still a bit iffy, so we wanted to do it ourselves to have exactly the same error spectrums and things in our sequences. And then we did a super simple experiment. So if we now start, say, with 2 Africans here, we can find all places where those 2 Africans differ from each other in their genome. And then we can ask for the Neanderthal, how often does the Neanderthal match a variant in this African or that African? And since there is no reason to assume that any African is more related to a Neanderthal than another African, since Neanderthals have never been in Africa, it should be 50-50. And this matching is indeed, statistically speaking, 50-50. Matches this and that African equally often. And then we did the interesting experiment then to look at a European and an African, see where they differ, and say how often does a Neanderthal match a European or the African? And to my big surprise back then, it was actually more matching to the European person. That looked very sort of compatible with that there would be a contribution from Neanderthals to present-day Europeans. It could still be contamination, right? But the most likely source of contamination would be Europeans, since most of the people working in our labs were Europeans. What was also surprising was this thing that for other parts of the world, if you take a person from China and from Africa, there's again more matching. Although most people say there were never Neanderthals in China. Papua New Guinea, everyone would, China one might discuss if there could have been a Neanderthal. There never been a Neanderthal in Papua New Guinea. We again find this more matching. So wherever one looks outside sub-Saharan Africa, you find this. So we sort of, with this analysis and some other analysis, we then suggested that there is indeed this contribution from Neanderthals to everyone outside Africa. And the idea that still to a big extent holds up is this thing that when modern humans come out of Africa, they presumably passed the Middle East where there were Neanderthals. And if they then mixed with Neanderthals there and sort of had babies, and those babies were successful enough to be incorporated then in modern human societies and had kids in turn, these modern humans could carry this Neanderthal variants with them out into the rest of the world, also where Neanderthals had never been. And the results seem to be that 1 or 2% of your genome, if your roots are outside Africa, come from Neanderthals. What has then happened is, of course, that we've gotten much, much better Neanderthal genomes. We now have Neanderthal genomes of 30, 40-fold coverage, where every position is covered many times. So we can see the 2 variants the Neanderthal got from his or her mother and father and so on, and look much more carefully at this. There are 3 high-quality genomes today from Neanderthals from different parts of the world. And we can look more in detail on this. And then the real thing that really shows this is not contamination from present-day people is that if we now look at different people today, and each line would be one individual here, and mark in red parts of the chromosome that is very similar to the Neanderthal in one chromosome, you will see that these are long pieces. It's sort of on average 50,000 nucleotides that are Neanderthal-like, sometimes 100, 200,000, sometimes a bit shorter. And that really shows that these are chunks of our chromosomes that then come from Neanderthals. And that then adds up to 1 or 2% of your genome. But different people will carry different fragments, as you will see. So you can say, if you go around in this room and try to see how much can you puzzle together of the Neanderthal genome by looking at all our genomes here, that sort of ends up being 40, 50% or so. So something like half of the Neanderthal genome is still around in people today. So if you like, they are not sort of totally extinct. They sort of live on in many of us today a bit. So an important early thing was, of course, to get Neanderthal variants from many different sites. One of them are very close to here, Cedron, where you work. And an insight that have come from looking at these things is that what's typical of Neanderthals was that they had very little genetic variation, presumably because they were very small population size. As we said in the beginning, Africans today have a bit more variation than non-Africans. If we look at here, sort of how many differences are there between two individuals in 10,000 bases, it's something like 2.7 outside and 3.5 in Africa. And you will see that Neanderthals had even less than that. So they were clearly sort of a small population size. We should see that we sort of accelerate a little bit so we don't get too slow here. Yes, there's this site in southern Siberia on the border between Russia, Kazakhstan, China, and Mongolia, the Denisova cave, where we got this tiny little bone that they had realized at excavation that it might be human. And it's fortunately very well preserved, so we could do a good genome of it. And indeed, it turned out to not be a Neanderthal, not a modern human, but something else here. That go back about 400,000 years ago or so, to common ancestors shared with Neanderthals. So this was sort of surprising. It was sort of, we had only a genome sequence and realized it comes from some other extinct group of human that we knew only then from the genome sequence. We didn't know how the skeletons look, what stone tools they used, or anything else. And we called them then Denisovans after this Denisova cave where they were found. And they have contributed to people today, just as Neanderthals have. So whereas everybody outside Africa have contribution from Neanderthals, we find no contribution from Denisovans in Europe or Central Asia. But wherever we look in sort of East Eurasia, we find a small contribution, less than 1%. And if you look in Aboriginal Australians or Papuans or so, we have 5-6% on top of the Neanderthal contribution. So there, something like 6-7% of people's genomes come from these other forms of humans that were here before. We can skip some of that. It gets boring. So we have only this genome sequence. We have few fragments of bones and teeth from that site that come from Neanderthals, from Denisovans. They had really super big teeth, but that's sort of everything we know about them. But we have this contribution all over Asia. So presumably they have been widespread all over Asia. And now there is one other site where one has begun to find something up in Tibet on the high plateau, where one found this mandible that has very big teeth, quite high up in the mountains. It's quite old, more than 160,000 years old. One have not been able to retrieve DNA from it. But the teeth looked a bit like the teeth from the Nisva cave. So together with Chinese collaborators, we studied that cave site. And what is now becoming more and more possible, that could be cool and Citron too to consider, is to study just sediments. That even if you have no bones, you can take sediments from cave sites particularly, and extract DNA from it and look what DNA you find there. So you then find animal DNA, you see what animals have been in the cave, and if you're lucky, you find also DNA from people that have been there. We really don't know where that DNA comes from. We don't know what they have done in the cave that have left their DNA there. You can speculate about what that is. But at this site then we have tested different layers through here, and we find then DNA in some of these layers, and that from this cave here we find DNA, mitochondrial DNA, that is related to this Denisovans from the Nisva cave. So we can show that Neanderthals have been in this cave and spat on the floor or whatever, peed in the corner, or I don't know what they did. So there is at least one more site in the world where we have direct evidence that Denisovans existed. And there must be many, many more. I would assume that many cranial skeletons that exist in museums in China, for example, will turn out to be Denisovans when one studies DNA from them. And it's actually an interesting area where one sort of begins to be able to retrieve DNA not from skeletons but from other things. Just as an example, in Denisova Cave in 2019, they found this pendant sort of that someone had hanging around their neck here. And fortunately, here, we got it before they had cleaned it. Normally when you find such things, you sort of clean them and get rid of everything. But we got it in this case. So we got to clean it in our clean room and extract DNA from the surface of it. And indeed, one can show that the human DNA in this sort of falls together with other Pleistocene humans from Eastern Eurasia. So at least in this case, one can actually retrieve the DNA from the person and show that it was a woman who had worn this, worn or produced it, who knows, but sort of that had handled it intimately. And I think that's sort of an area, hopefully, that comes more in archaeology. That will be very interesting when you can retrieve DNA like this and get the specimens directly from the excavation to see, was it men or women who handled this thing, what population did they come from, and things like that. Just to wrap up then, this part, modern humans evolved in Africa, they come out, they meet Neanderthals, they meet Denisovans, they mix early on, they mix with Denisovans in the east. And, oh, that's sad, no map anymore. Ah, there is no map. We have these contributions today. And we begin now to learn more about this replacement. So what should we talk about? We could talk more about this replacement history, or we can spend more time talking about the influence of genetic variants from the Neanderthals, particularly in present-day people, and what consequences it has medically and things like that. We don't have time to go through everything I have here, I think.
I think that they will be more interested in how those variants affect
present-day people. Okay, people are nodding. Acceleration.
I mean, everything is related.
Yes, but yes. So an introduction to that sort of is to say then that when we now have high quality genomes from our closest relatives here, and we have, as you know, hundreds of thousands of good genomes from present-day people, and we have the apes, we can sort of begin to see where genetic changes happened here. Right? So when we find something that exists in present-day people and in Neanderthals, but not in the apes, it's a change that happened back here and spread to everybody. If we find it only in Neanderthals and we look like the apes, then it happened here on the lineage to Neanderthals. And if it exists in everyone today, or almost everyone, and not in Neanderthals or apes, it happened here. So we want to illustrate some examples of this to you and sort of start with something here on the line to Neanderthals. And we now have 3 good Neanderthal genomes. We have like 12 lower quality ones. So we can really begin to look at what seems to have been there in all Neanderthals. And there is a person, Hugo Seberg, who works with us, who is particularly interested in ion channels. Some proteins that sit in the cell membrane and sort of regulate how ions are pumped in and out of the cell. And he noticed that if you look through all the ion channels in the genome in the Neanderthals, there was one ion channel, this one here, now 1.7, that had 3 amino acid changes. And it really stood out. It's the only ion channel that had more than 1 amino acid change in Neanderthals. And it's there in all Neanderthals we looked at. And humans doesn't have this, we thought at the time anyway. So that looked interesting. So what we did was to produce this ion channel and a modern human version of the ion channel that then differ in these 3 amino acid changes, and inject the mRNA for them that we had synthesized in vitro. Well, not so fast. This ion channel is interesting because it sits in the peripheral nerve endings in our tissues, and it's involved in the initiation of sense of pain. So when you hurt yourself, it's sort of this sodium channels through the sodium ions through the membrane and initiates a sense of pain. And so what we did was to sort of synthesize the mRNAs for these 2 versions of it and either inject it in frog oocytes or inject it in human cells and study the electrophysiology of this. And I'm not an electrophysiologist, but they have explained this to me, that if you sort of stimulate then cells that express either the modern human one in blue or the red one in Neanderthal one, you sort of seem to have a bigger current through the membrane with the Neanderthal ones than with the modern human ones for the same stimulation. So it sort of seems to be more sensitive, if you like. And you can also show that that is not due to that it opens faster, but that it remains open longer once it has opened this channel. Because after it has opened, there is sort of an inactivation that happens when the ion channel is sort of refractory and cannot be stimulated anymore, but that is less efficient for the Neanderthal version. And you can then, we also synthesize then all combinations of these 3 amino acid changes. So you could show if you just introduce one of the amino acid changes, there's no difference whatsoever to the present day version. But if you introduce 2 of them, these 2 ones, you have this effect of a more sensitive channel. And it's the 2 intracellular amino acid changes there that sort of do that. So that seemed interesting. And as I said, we thought this was specific to Neanderthals. But what's also happening in the world is that we get more and more present-day genomes. And what is particularly interesting for us is, of course, when we have the genomes of people and we have medical records and we have questionnaires and know something about these people. And the biggest such biobank that is accessible and you can work with easily, fairly easy, is the UK Biobank in Britain. So there were then like 360,000 people there where you have genome information, and they have answered lots of questionnaires and know a bit about their medical record and things like that. And when we looked at that, we actually found that there are some people today that carry this Neanderthal version. So 0.4% of people carry this. So we got very excited because it's opened a way, of course, to study this variant in a relevant individual. So can then use modern humans, as you like, as a sort of experimental animal to study sort of Neanderthal versions of things. And in this case, we then looked at all questions that people had answered that had to do with pain. So all kinds of pain, headaches, stomach pain, back pains, what have you. And I think there were 17 questions there that had to do with pain. And it was the first time for me when I could sort of play a bit with such big population data. So sort of the first question is, what does sort of pain in your life correlate with mostly? And it's very sad when you get older. Age is the biggest correlate. So the older you get, the more pain people report in their lives. So we did very many politically incorrect questions. We asked for men and women, how is the sensitivity to pain? And fortunately, there is no difference. I don't have that here, but there's actually no difference. Otherwise, it would have been sensitive. But more relevant then, we could ask about the carriers of this Neanderthal version. What about them? And don't forget, they are heterozygous, right? This is so rare. We have 2 chromosomes, of course. And if you have something that, 0.4% of people have, all the people have 1 chromosome, is a Neanderthal, one is a modern human. But nevertheless, these people actually reported significantly more pain in their lives than people who didn't carry it. And if you now, to get a feeling for how big an effect this is, if we relate this to this age effect, if you're a carrier of this Neanderthal thing, it is as if you were 8 or 9 years older in terms of how much pain you experience. So it is sort of some effects that's actually a bit really there. Of course, we all know that, we cannot say Neanderthals experienced more pain because of this, really. We all know that the sense of pain is modulated in the spinal cord and very much in your brain, right? Even the same stimulation can sometimes be pleasurable and sometimes painful, dependent on what you're doing. And the other thing is, of course, what I also said, that all these people we have here are heterozygous. They have the Neanderthal on 1 chromosome version and the modern human one on the other. But nevertheless, it's sort of interesting. We have this view of Neanderthals, and Neanderthals had it on both chromosomes, yes. But we have this view of Neanderthals that they were this sort of insensitive, brutish people. But maybe they were actually more sensitive. Maybe it's actually that they were hypersensitive, and when modern humans came, everything hurt, so you become extinct. I don't know. But sort of, one begins to learn a bit about Neanderthal physiology from this. Shall we stop for questions at some point? Someone has a question? It's embarrassing. I just talk and talk. Now we come to the next variant here.
I don't think they want to interrupt you.
Are there any questions about this? Yes. Oh, now you really intimidate people. Now you get on television because you...
Okay, it might be a dumb question, but when you were talking about Neanderthals having a very low variability between them, I came up with the question like, do they have low variance because maybe we're finding individuals from like at the end of their evolutionary cycle? Like maybe they didn't have a lot of variability because they were about to be extinct, or because that was just their biology and their behavior?
No, that's a good question. I should have said that. I have to be close to the microphone, so I sit down. Yes, of course, when you have a small size, and one time you may have a small size is when you're becoming extinct, right? So that could be true for this first Neanderthal. That's probably a very late Neanderthal. It's something like 45,000 years old, and this is sort of when they disappear. But we also have this a high-quality Neanderthal genome now from Altai site, and that's at 120,000. So that is sort of like whatever, 70, 80,000 years before they become extinct. And we begin to have variation from within the groups. They seem to have lived in small groups, which sort of make for little variation. And it's in general an interesting thing. Sort of, there's an sort of feeling around that if you have a little variation, that's very bad for you. And people talk about inbreeding, which is sort of a bad word. It sounds like you're sort of dumb and I don't know. It's not always the case that species or groups with little variation really do badly, actually. There are examples of where that is, and it depends on the variation that's there in the population, I think, right? I think it's particularly bad if you're a big population with much variation and you shrink. Then you may have many recessive lethal things there that sort of then become homozygous and you have many problems. But if you're small size over a long time, those things disappear out of the population. And then you're fine. If you established inbred lines of mice, right, that you have in your experiments, they have no genetic variation, and they are fairly okay. When you sort of establish, some of them really have problems too, but you go through a crisis when you establish those mice, where many of them die and you may lose them. But when you come through with that and have purified out those things, you're doing well again. And there are species in Northern Europe, such as elk and beavers, that have over very few 100 years ago and now have expanded and are doing very well. So this is sort of a tangent, but in conservation genetics, it's often said that it's so bad when you see little variation, but it's not always true, I think. And I don't think that's why, sort of in this case, it's... How much time do we have? How long should we go on?
You're 1 hour.
I don't know how long you got. Maybe, I don't know. So just another example of something in Neanderthals then has to do with a hormone that many of you know about, progesterone, and the receptor for progesterone. So this is a sort of a steroid hormone. It goes into your cells and it has a protein receptor inside the cell. And it's a receptor we're interested in. And it's an example of these things. It's a sort of a variant exists of the progesterone receptor that is known to be associated with premature births, that you have your baby too early. And that was known way before the Neanderthal genome came around. But what was always suspicious or sort of interesting was that this risk variant then for giving birth too early exists hardly in Africa at all. It exists in Europe and Asia and in Native Americans. And now we sort of know that whenever you see a genetic variant like this, it almost always comes from Neanderthals. When it's sort of absent in Africa and exists outside Africa, it's almost always an indication that it comes from Neanderthals. And it turned out to be the case. And it's then associated with preterm births. And people have really, when they found this, sort of speculated that this is sort of weird. It's probably really a bad thing for Neanderthals. It may have been a disadvantage contributing to that and disappear and things like that. Lots of speculation. Evolutionary research is full of speculation. You should be careful with that. Storytelling. But a thing that now becomes really important, I think, is that we have not only Neanderthal genomes and Denisovan genomes, but much, much more. We have tens of thousands of genomes of modern humans distributed in time over the last 30,000 years, particularly in Europe, where much of this work has been done. So this is not up to date. Now we have something like over 20,000 genomes from, say, 25,000 years ago to medieval times in Europe. And the cool thing is that we can then follow over time how genetic variants has changed in frequency. So we don't anymore just have to make models, look in present-day people, and try to infer what was positively selected, what was negatively selected in the past. We can actually go and check. And as often, many things that we thought turned out to be wrong. You know, this lactose tolerance, for example, is a sort of textbook thing that people in Northern Europe particularly can drink fresh milk without getting diarrhea, and in many parts of Africa you can't do that, and that must have come when we started dairy farming and started having cows. Now, when we actually go back in skeletons and look when people do that, it came much, much later. So it was at least 3,000 years after you started with the cows. So sort of many of these stories you read in books are actually then not true when you start. But in this case then, one can go back and look, particularly in Europe. And here we made a little animation to show sort of what happens with this variant. So we start 15,000 years ago. The red ones are the Neanderthal Denisovan genomes. Carriers of this are black, of this risk variant for premature babies. Non-carriers that don't have it are gray. And we just go forward in time now and see what happens with the frequency of this. And when we get to 7,000 or so, you will see that this thing almost explodes in frequency, which makes no sense whatsoever. I mean, particularly when you don't have good medical care, having premature babies is a huge, huge risk to the baby. So sort of, how could this be? How could that increase in frequency? But that made us go back then to the UK Biobank and look in present-day people, carriers of this and the modern human variant, what about them? And this is a bit not a good slide. Here we look now at the modern allele, the non-Neanderthal version, the ones that's protective against premature babies, and see what is associated with today in the present-day population. So that variant that protects against having premature babies is actually, though, associated with bleeding early in pregnancy, with miscarriages, with losing your baby, and it's negatively associated with numbers of full sisters and also brothers, but not quite significant. As you have, if you have the modern version, you have less brothers and sisters, you have more miscarriages. So it seems to be a much more complex story than what one thought. This Neanderthal progesterone receptor, yes, it is associated with the risk of having your babies too early, but it is actually protecting you against miscarriages, of losing the baby, and results in more live births. So it's probably really an example of a sort of trade-off that having this version saves pregnancies that would otherwise result in miscarriages. But the price you pay is that some of these babies are born early, but they're at least born. And we begin now to understand why this is too. But because if you look at this variant and look at the expression of the progesterone receptor, you find that if you express this one, if you have this Neanderthal version, you express more of the progesterone receptor. So this is an EQTL, if you like, an expression QTL for this receptor. So you could imagine you have more of a progesterone effect with the Neanderthal version, and if you have more receptor here. And very satisfactory in the last 4 years, there has been 3 studies where one has given progesterone, so give more of the hormone, to women who has experienced miscarriages previously, and you can then increase the number of live births quite substantially. compatible with this idea that more progesterone protect risky pregnancies. So we're beginning to understand, and you can do that presumably by upregulating a receptor or by giving more of the hormone. And it's a sort of pattern that many variants that are known medically turn out to come from Neanderthals. So there are 2 cytochrome P450 enzymes that are expressed in the liver and are involved in digesting many medicines, metabolizing medicines that you take. They have variants that come from Neanderthals. So for example, if you take ibuprofen against pains or so, you have a much better effect if you have this Neanderthal variant, because it slows the metabolism, so you have a higher dose for a longer time in your blood. But the other variant then metabolizes warfarin that you take against blood clotting if you have had a stroke or you have an artificial heart valve, for example. And there you don't want necessarily higher effect because you may have bleeding as a side effect, so you need to take a lower dose if you're a Neanderthal carrier. So sort of have effects present day. And the biggest shock we got was in the pandemic, where, as you know, we had this SARS-CoV-2 virus, and most people have no symptoms or very mild symptoms, but some people become severely ill and even die from it, particularly early in the pandemic. We know some of the risk factors, old age, male sex, diabetes, what have you, but all that is not enough clearly to explain why some people become so sick and some have very little symptoms. So there was clearly that there were sort of genetic factors in people too that influenced this. So we were a bit peripherally involved in a big consortium, international consortium, to study people who were hospitalized and try to find genetic variants influencing outcome when you're infected. And early in summer of 2020, the first results came, and it was very striking. There was sort of one variant on chromosome 3 that had a huge influence on this, a very strong effect for being a genetic risk factor. Otherwise, often when you look at this, you have a little things here and there that all contribute. It was one big thing that influenced this on chromosome 3. And we were like totally shocked when we looked at this, because if we then look at these risk variants here, they are closely related, particularly to one of our Neanderthals, the one from Croatia. And all the protective ones that sort of decrease your risk of becoming severely ill are sort of very far from the Neanderthals. So this risk of becoming severely ill clearly comes over from Neanderthals into present-day people and exists in some of us today. And this is, as I indicated, quite a big risk, quite a big effect size for being a genetic risk factor. So if you were hospitalized in Scandinavia, the first year of the pandemic, so severe so you were hospitalized, you had a 7% risk of dying if you were not a carrier of this, and your risk of dying were about twice that if you have the Neanderthal version. So it's a substantial risk. It's sort of scary. It's sort of, you almost see selection in action, right? If you have a genetic variant where it doubles my risk of dying, it's sort of, that is what selection is. So, of course, there were great interest in what is in this region, what are the variants there. There's sort of a 50 kb region that defines this. And we and other groups worked like crazy on this, and unfortunately it's a very complex region. There are several genes in there. There are at least 3 genes where you can make plausible stories about why it would be. There are 14 nucleotide differences between risk and protection, and one hasn't been able to figure it out yet. I mean, eventually nothing changes in amino acid, some things change expression of some things. If we understood this, one would presumably be able to treat perhaps severe COVID also much more efficient, in particular the ones at risk of dying. But there's other, so one could then, this is the frequency of this around the world, as we see it, absent in Africa, but sort of existing outside, but at very different frequencies, right? But you can now use this frequency to estimate how many extra deaths did we have due to the Neanderthal contribution, and it's at least 1 million extra deaths. So it's sort of, one shouldn't joke about these things, but it's sort of the late revenge of the Neanderthals, right? There were probably 200,000 Neanderthals that became extinct, and now more than 1 million people have died due to this Neanderthal contribution. But you also see something interesting on this figure here, and that is this difference here. It's sort of absent in East Asia. In China and Japan, this thing does not exist. And it's very frequent in South Asia, in India, Sri Lanka, Pakistan. It's actually, if you look genome-wide, it's the sort of most dramatic frequency difference between India and China that you have. So clearly there has been something going on. There has been negative selection in China for this in the past and in Japan. We don't know what that is. It's of course tempting to speculate that there might have been epidemics of coronaviruses there or something like that, so it's selected against it. But it has clearly been positive in China to carry this. We also have no idea what that is. Presumably protective then against some infectious diseases and so on. We're beginning to learn a little bit about that this variant, the risk variants here, influence other things in our biology. So Hugo again, he's sort of, if you look at this risk variant on chromosome 3, if you look 1 million base pairs downstream in the direction of these genes here, downstream of this, there's another gene there that is very famous. Some of you have heard about it, I suppose, CCR5. That's a co-receptor for HIV, for the virus that gives you AIDS. So HIV uses this as a means to get into the cells. And it's a famous gene because in the early epidemic of HIV, one found that there were some people who were clearly exposed to the virus but never got infected. And it turned out that these were people who lacked this gene, who had a stop codon in both versions of it. So if you lack CCR5, you don't get infected. But if we look now at the people who carry the COVID risk variant for their expression of CCR5, you find that they express less of the CCR5 gene. So this risk variant here influence expression of CCR5, it's an EQTL for CCR5 and downregulates it. And if you look in cohorts, in 3 different cohorts, in the UK Biobank, in a Finnish cohort and a US cohort, of your risk of becoming HIV positive, it's sort of reduced if you carry this COVID variant. So it's sort of quite interesting. This sort of increases your risk for severe COVID by doubling or almost tripling it if you get infected. But if you're exposed to HIV, it actually reduces your risk of being infected. So it sort of really shows this thing, I think, that genetic variants have their effects influenced by the environment. They may be good in one certain, if you're exposed to HIV, it's really nice to have this variant. If you're exposed to COVID and get infected, it's really bad to have it, right? So it's sort of a double-edged sword, the genetic variants, and really often many effects depending on, this HIV is perhaps 50 years old, 40 years old. COVID is 5 years old. These things that change the frequency in China and in India are clearly other things, right? So it must have other effects. It's sort of a multi-edged sword, these things. I think it's sort of also good to think about this, about when you hear people talking about sort of that certain variants are bad or one should sort of modify the genomes of people and things, we sort of understand so little of this, right? One could say if we only had that COVID information, sure, we should get rid of that variant, right? But then in other situations, it might actually be really good to have it. So sort of, we know far too little about these things. Yes, there are other variants. Now we have so big cohorts, we find some other variants influencing severe COVID, some Neanderthal variants even slightly protective, but the effect is way smaller. Right? This is the big effect there that we talk about. Well, and before wrapping up then, there are things here. That's perhaps the thing I'm most interested in now. Things that change on the human lineage and are there in almost everyone today. And it's sort of interesting because modern humans are very special to my mind. I don't know, we could discuss that. But Neanderthals existed longer than modern humans have existed. They never became millions and billions of people. Their culture did change, but on a very slower time scale, is my feeling, than modern humans. At least since 70,000 years ago or something like that, modern humans have a culture and technology that changes really rapidly. So technology, art, that would be an interesting discussion. What art did Neanderthals have? I tend to be a bit sort of make fun of it a bit and say that, yeah, in Spain here one has found things that are dated and should sort of be Neanderthal art. When I look at it, I say that's very modern art because I can't see what it depicts, sort of. Whereas sort of art that's really figurative, where I needed to see what it is, that comes with modern humans and becomes very common. And people become very numerous. They spread over open water systematically, right? Come to parts of the world where humans have never been before, etc. And the feeling is that it might be an illusion, but sort of a feeling thing is that there might be some genetic, there's of course not genetic variance determining your culture, but that there is some genetic variance that makes us able to absorb culture quickly as kids and sort of adapt it and sort of, so there might be something here. And we can of course look at these variants now. We can look for things since we have this genome that is in everyone or almost everyone today. And it's not that many. So if you compare two genomes between two people, or the genome you got from your mom and your dad, it's like 3 million differences or so. But if you make the condition that 99.9% of everyone today should have a variant, and Neanderthals don't have it, it's like 30,000 differences, so like 100-fold less. So it's not that many. You can scroll through those differences in an afternoon in a computer. The problem is you don't know what you'd look at all, because another part of this is that we're incredibly bad to just, from a genetic variant, say what consequences it would have, you know. Everything we know almost comes from association studies that we discussed, where you look in population and see what variant associates with something, how you do when you get infected with COVID, or how much pain you feel, or something like that. But if you look for things that everyone has today, it's sort of very hard to study that. But we're sort of interested in that. And we have this bias, of course, that when I list what's special for modern humans, it has to do with cognition or behavior and things like that. So just as an example, what we work at, we do one example, this is a list of all proteins with amino acid changes that almost everyone carry today, and they're not there in Neanderthals. And that's about 87 proteins. There's not many. But what was very striking to us when we looked at this was that 3 of these proteins are part of the machinery that pull chromosomes apart during mitosis. There is this metaphase plate that is formed, and the spindle and the kinetochores, and these are proteins involved in the kinetochore and spindle. And not only that, this is the only protein on the list with 3 amino acid changes. That's one of the few of them with 2, and that has 1. So 6 of about 100 amino acid changes have to do with mitosis, which seems totally crazy. I mean, if I would say something that I would have thought would be very conserved and not have changed in 500,000 years of evolution, it would be cell division. But so be it. But sort of, these genes are involved in this, and it made us think then back on a study back from 2016 when we used brain organoids. So these are, you take stem cells and you can differentiate them in tissue culture to little balls of 1 or 2 millimeters that sort of mimic early development of the brain. And we did this together with Wieland Huttner's group, comparing human and chimpanzee early brain development then. And what found hardly any differences, of course, between how chimp and human brain develop. But the only difference we really found was if you look at the chromosomes and the DNA stained here in a little movie, don't know if you can see it. If you look here in a chimp, you see the chromosomes condensing to a metaphase plate, they're lined up, and here, poof, they're pulled apart. And in humans there. And as you saw, in humans it happens slightly later. So this phase when they are lined up here together and the cell makes sure that each one chromosome will go to one daughter cell and one to the other daughter cell, that it really functions properly. It's longer in humans than in chimps. And since now sort of we have these 3 genes with 6 amino acid changes in encoded proteins, thought maybe that has to do with it. So we made a mouse where we engineered in these 6 changes and these 3 things and looked at that, the cell division during brain development, exactly like that. And strikingly, if we look at metaphase length, it is indeed longer when we have these changes in these 3 genes or in 2 of the genes. When we do them singly, we have no effect or some intermediate effect. So these 2 genes seem to be important for this, prolongation of metaphase. So we then go to human stem cells and with CRISPR-Cas9, edit them back to the ancestral state, and then we have this shortening of the metaphase there. So it seems to be due to these changes in these 2 genes. Does it have an effect? So sort of this difference between humans and chimps that we know from before have really happened recently here, since we separated from Neanderthals. So does it have effects? Well, you can sort of look at missegregation of chromosomes. In how many of metaphases you observe is one chromosome lagging or goes to the wrong place. And there seems to be an increase here between these human organoids and the ancestralized organoids. It's small numbers, it's bad statistics, but it still sort of goes in the right direction. So it sort of seems to be that something have happened here in how accurately cells divide when the brain is formed. So, but we don't know if that has effects on in the adult at all. So we're analyzing the mice actually, see how smart the mice are, the mice that are humanized from these. In intelligence tests for the mice to sort of see if they are better in anything. And there's another gene we're also looking at, but we're getting tired of all this. It's a big question sort of with this and another gene is there some difference in how neurons are formed when the brain develops? So sort of that's really where we are. That is sort of, we've sort of, my own group has sort of left doing ancient genomes and studying more the effects of these things now. Yes. I don't think we need to look at all this. In the end, sort of, it's that being a modern human, what makes you a modern human, I think is really a combination of things. It's not going to be we find one gene and one change and that is it. It's going to be a combination of things. Even this effect on the segregation is due to 3 different changes in 2 genes, right? And I think some other things we're interested in is going to be even more complex and involve more genes. So yes, that is sort of what we do. We study these things, we study contributions, we use biobanks a lot, and stem cells and editing and humanized mice and things like that. And that is the sort of places we do this. Most people are working in Leipzig in this group, and a small group in Okinawa too, where there's good snorkeling and diving. So now there must be some questions, I hope. Yes. Mm-hmm.
Uno, sí. Thank you for your talk. I have two maybe personal questions. Is allowed? One is about, recently it was the first child that was born after a genetic screening. There are some companies now that offer to sequencing the complete genome of different gametes and then make some kind of selection on the gametes, no? We are now in that era, no? That it's possible to make complete sequencing of many genomes, and there are many people, even scientifics, relevant scientists that say that they have their genome sequenced, and there is that possibility today, no? And I want to know about your opinion about personal genomics. About all this development that is today and that databases and who has that data and to make work. What is your view on that, about the ethical or problems that can be associated with that? That's I think that one is enough.
Yeah, I was about to say there are many aspects of that what you ask about, of course. Say personal genomics, in a sense, I test myself. I would only be interested if it's something that I can really result in something I can improve in my sort of behavior or something, you know. If I turn out to be homozygous for APOE4 and have a high risk for developing Alzheimer, there's nothing I can do about it. So do I want to know about it? I probably prefer not to know, I would say. Whereas, yes, if there are certain things that are, but many of these things, but I'm not an expert on this, but my bias personally would be to say many of these things where you now use polygenic scores, where you look at variation all across the genome and say, what is my risk for arteriosclerosis or for colitis or for something? They are still so bad, so that I would not really. I would say it's a little bit, we kind of know how to live a healthier life, and that's what we can do. We know we shouldn't smoke, and we shouldn't get too fat, and we should move around somehow. And then it gets even, the total other aspect is, of course, yes, if you look at unborn babies or something, there are, of course, some things that are really so severe that one can reasonably consider to terminate the pregnancy because it's sort of really, really very terrible diseases and very short life. But that then becomes very personal, and many things are more in a gray zone, like trisomy 21, the most common one, for example. You can still have a satisfying life, I think, but it's sort of, yes, there are many. I think it's really personal decision, right? Yeah, yes.
Thanks for such an amazing talk, and I'm interested on that. So far you present some of more monogenetic trait. If you have some examples of the effect of adaptive integration in more polygenic traits that I know it's a little bit more difficult to
Yes, of course.
find.
Sort of old-fashioned in the sense that I like to study something that has a strong enough effect that we can show it in the population studies clearly, or even in experiments in the laboratory. Of course, almost all these things are polygenic. I mean, even the severe COVID there, as I indicated, we now in bigger cohort studies have more effects. There's one variant of chromosome 12 from Neanderthals that actually is protective and reduces your risk with 15-20% of becoming severely ill, whereas this other one, doubles or triples it. So almost all these things are polygenic. I think that there is, what you can do is, of course, look in polygenic scores for complex, really complex traits and see how much of the contribution comes from Neanderthal variants. And that is sort of, yes, you can do that. You can even do that for things like educational attainment and things like that. It gets politically a bit sensitive rather soon. But that is just an area that one would. I think some of these things, I mean, it's a very interesting thing generally in biology that I think we've come to realize, you're probably more aware of this than me, that many of the traits we are interested in are very polygenic and may have shifted just by rather quickly over 1,000 years or something by shifting allele frequency slightly at many, many loci. And even height or things, there are gradients in Europe, again, body height or pigmentation and things like that, that has obviously changed quite rapidly over, 3,000 years. And they are polygenic. So the idea is that they, if you shift 100 loci, just 1% in one direction, all of them, in a population, the trait will change quite dramatically. So I think a lot of this and many of these modern human-specific things may be like that too in the end. And it's sad, in a way, for genetics. I mean, it has been almost oversold, genetics. It's saying we will understand so much, but when it gets so polygenic, it also becomes untractable. You don't have a drug target really then, you don't sort of, what can you do? You can do nothing. But yeah, I don't have any much of an answer.
First of all, it's an honor to meet you. This lecture has reminded me why I love science, so thank you very much. And when I was listening to you and talking about the contribution of Neanderthals to modern humans, a question came to my mind. Why do you think the contribution was only found in nuclear DNA and not in the mitochondrial? Because it is like if only male Neanderthals have contributed, no?
Yes, that is a sort of a possibility, yes. The thing, we were very excited when we got the first Y chromosomes from Neanderthals, and it turns out they haven't contributed Y chromosomes either. The sort of the population geneticists that have modeled this, particularly someone at Berkeley, Monteslatkin, has sort of suggested that this is still compatible with just that there is a much more of chance events when you contribute mitochondria or Y chromosomes, because in a pair of humans that have a baby, there are 4 genomes, nuclear genomes, that can be passed on, 2 in the mother, 2 in the father. There's just 1 mitochondrial genome that will matter and 1 Y chromosomal genome that will matter. So it's much more of a chance. If a woman has only male offspring, her mitochondria is dead. And if a man has no sons, his Y chromosome is dead. So it's much more rapidly lost in the population. And that's still compatible with that it's just such a chance event. But there's another observation that's on the X chromosome. There is actually less Neanderthal contribution on the X chromosome than the autosomes. And that's also compatible when we since females contribute 2 X chromosomes and males have only 1 X chromosome, it's sort of compatible with that it would be more modern, that would be more males, Neanderthal males, I get this right now, would be more Neanderthal males contributing because the Y chromosome, the X chromosome contribution is less. Now there are other parts on the autosomes too where we see less contribution because of selection, presumably, against Neanderthal versions. And you can also argue that on the X chromosome there's more selection because for the males they have only 1 version of the X. So if you have something slightly bad on the X chromosome, as a male it may be really bad. As a female you may compensate that by your rather good X chromosome. So it might just be that there's more selection going on the X. It's sort of, probably really that's why also this X effect, have nothing to do with Neanderthals, but there's a real observation that there are a lot more mental retardation in males than in females. If you look even at classes of speech therapy in the school where my kids go, there are a lot more boys and girls who need help learning to speak. And probable explanation is that for 5% of the genome, males are really slaves of the 1 chromosome they got, right, and have no heterozygosity. But we don't know this answer. We don't know.
Hello, so I join everyone who has talked in saying that I'm really grateful for this lecture. And to be honest, my question is a bit speculative, and I'm not that proficient in anthropology, so if there's something terribly wrong, please correct me. But when you were talking about all the different variants you have found that were like found more in the Neanderthals, like the more sensitive to pain, the risks of premature births, and the more vulnerability to certain epidemics, I was thinking about maybe it has to be with the social structure. Like, I think that in modern primates, those who live in smaller groups tend to care more about the members of their group, while those who live in larger groups tend to be like more on their own in that sense. And maybe those variants, like if you are more sensible to pain and you are going to be unable to care for yourself when you are older, you will have someone to care for yourself. If you have a premature baby, you will have help in raising that baby. You are like wouldn't be positively selected to be resistant to epidemics since you are in a small group. And I think that I was wondering if you think this could be a case of co-evolution and a glimpse at how the social structures we build affect our evolution, and if maybe we'll need to start thinking about it not only as scientists but also as a society.
Yes, so there could speculate in different directions there. Yes, I think it's sort of evidence that Neanderthals lived in smaller groups and their overall size was less. It's true that in smaller groups, slightly deleterious variants can accumulate more easily. Also just because of, how genetics work, that sort of selection. If there is slight selection against something in each generation, if you have a huge population, before it becomes fixed, it has to go through many, many generations, so selection is sort of stronger. Whereas in a small group, there are chance events that things that are slightly deleterious can become frequent. So that's one thought. That's sort of one idea why there might be slightly deleterious things in Neanderthals. And another thing that comes to mind is that we don't know if variants are bad or good in many sense. Being a bit more sensitive to pain may actually be very good. You may react more quickly to something that's bad for you. This variant there, this ion channel, there are people who lack it today, and their life expectancy is very short because they don't sense when they get sick or even when they hurt themselves and things like that. So it's actually good to be sensitive to. But yes, otherwise the sort of things I would sort of defer to saying we should think about how we want to live our lives as human beings and take care of each other. And biology has nothing to say about that, I would say. I think it's sort of an ethical, political thing that I think it's almost dangerous to say that evolution and genetics should have a role at all in that debate. We kind of have agreed that you have human rights irrespective of your biology, I would say. So going in that direction is almost scary to me, and the ideologies that have done that in the last century sort of really went bad. So.
As you can see, students are asking, so if they didn't ask before, it was because they didn't want to interrupt the talk. It was, of course, a wonderful talk, and I will have to restrict myself to just some questions. We tell students that nothing in biology makes sense if not in the light of evolution. And I think you have shown us evolution happening. And there's this thing also related to what they were asking. We are making, with these Neanderthal things, we are making quite a long experiment in decoupling regions of the genome. Like a very important part of evolution that Dobzhansky already talked about more than a century ago is that sometimes you need to be able to acquire something negative that together with something else becomes positive. And that something else may be far away in the genome. Now we have uncoupled all of that. We are just inheriting one part, not the others. And perhaps that's also a piece that is lacking in understanding. Like you yourself said, we cannot know if Neanderthals actually felt more pain. Mm-hmm. They might have something that was fixed at the same time, and now we don't have those two things together. So we are making that experiment, right?
Yes, and our dilemma is a bit that we don't have. There's probably at least 40% of the Neanderthal genome we don't have as a contribution today.
But even the parts that we have, the same individual does not have the same part. So
but for the part we have, I think I'm very optimistic about this thing that we are getting biobanks now, not only in Europe, but in Asia and in the future in other parts of the world, with eventually millions of people, that we will find these rare combinations of things if they exist. And I actually think that, a dream would be to have biobanks in Papua New Guinea or something like that, where there is sort of much larger part of the archaic variation exists. So I'm rather hopeful we understand some of that, at least. But yes, it's certainly true that it's...
So just a more direct question. You were talking about those variants that were fixed in the human population after let's say we found those Neanderthals. But that's a fixation that happened all over the world, right? Like in all continents independently. Wouldn't that tell us that there was some kind of event that changed and there was some selection?
Well, I would say that this fixation for these 30,000 things that are not that many, they probably happened, I would guess, early on in modern humans in Africa before one spread out. I would say from 30, 40,000 years ago when one were already spread out, then probably we don't have fixations anymore. So,
which is what you would expect because we are really far away and not communicating anymore. But also in Africa they have those.
Yes.
So it kind of happened.
yes, of course, modern humans evolved somewhere in Africa, spread across Africa just as in the rest of the world, and replaced these other forms. So sort of modern humans are really very special, I think, in the sense that they have replaced everyone else. And until 30, 40,000 years ago, there were always different forms of humans around on the planet. And the last few 10,000 years are really unique in that we're alone, which is sad, perhaps. Mm-hmm.
Maybe I could do a practice. Hazlo tú primero.
So thank you for the talk. Now we're with the questions. I got another one. So every example you put in the table, it was like protein coding DNA. How about non-protein coding DNA we have heritage from Neanderthals? You know, like having a higher level because we get a promoter or something like that. I don't know if there is any study about that have this in mind.
Yes, there are studies, and sort of we have been lazy with this modern human thing, so saying we start with some amino acid changes because it's somehow easier. We can synthesize the proteins, we can study it. If it's an enzyme, we even studied enzymes, I didn't talk about that, where you can say, we know what the protein does, it catalyzes this reaction, and we can study that in vitro. And some of the things, that, like the COVID risk factor is an expression thing, you know, there's no structure changes in the genes there. There have been studies of studying promoters and enhancers, not by us, by other groups, Josh Akey's group, particularly at Princeton, looking at influence on expression of neighboring genes from Neanderthal variants. And there are some interesting observations there. For example, that they look at people who are then heterozygous for having Neanderthal and not, and where they can look at by linkage on which allele is expressed higher. And in certain tissues, it is very striking that the Neanderthal versions are expressed less. And the brain is actually one such tissue, and the other one is liver, I think, where there is a big unbalanced expression. So it is as if there's sort of less favorable, you sort of selected the Neanderthal version that's expressed less. So there are studies like that, yes. To study expression.
I am going to do maybe a practical question, because here we have a lot of students, maybe people who's doing the PhD, people who have finished. And you know that Svante work in the Max Planck institution that I think is one of the most important in the world. Many reasons. So maybe it will be useful, you can explain the students and people, what's the way, for example, to go to the Max Planck Institute to work in the Max Planck institution, to work in the different institutes, what they have to do, you know. Study, sure.
Nothing special, I mean.
Select the institute, surely.
Yes. I think the most important thing that has nothing to do with Max Planck, I mean, I think the almost most important thing is choosing a good advisor and good lab to be in. And yes, the Max Planck system has its advantages and has its problems also. I think it's very focused on a PI, on a principal investigator. So even more than at some universities. So it's sort of, if you're in a good functioning group and department, that's great. if it is bad, it can be very bad because it's so dependent on one person. I think the most important thing if you're interested in science is, yes, where do you go? Both is that a group that does good science, publish well, and is well funded? And is it a place that works for you from a personal point of view? It's after all still a little bit like being an artist and working with someone teaching you art or dance or something. You must get along personally also. This sort of must function. But otherwise, yeah, I don't know what to say really. Visit in person and go out and have dinner without the professor there. So you get a feeling for how it actually works in a group. Maybe. Mm-hmm.